artificial gene, multiple cloning site, both A and C. Now, we are aware of vectors. Vectors are nothing but the DNA which carries a DNA of interest, or it carries a foreign DNA which is of interest. That is, it produces a protein which we are interested in. So, such DNA which helps in carrying a foreign DNA is called a vector. So, here we are talking about a vector in which number of recognition sites are arranged together in a particular area. So, basically when we see vector, there are certain areas which are very important to be present in the cloning vectors. For example, they should possess ori, or the origin of replication site, for replication to take place. The other thing which is necessary is selective or selectable markers. It can be some genes which will help to find out which are the transformed bacteria. And another very important thing which is required in a vector is presence of restriction sites. Now, if a foreign DNA has to be inserted into a plasmid, the plasmid has to be cut open, what we call cleave, or it has to cut open so that in that place we can insert the foreign DNA. So, we use molecular scissors, what we call REN, restriction endonuclease, for this purpose. Restriction endonuclease cannot just go and cut in any point of the DNA. It recognizes some unique sequences which are called restriction sites. It can cut only in those areas. So, to be a functional vector, what the vector requires to have is one more site, that is the recognition site. So, when the respective restriction endonuclease is used, it will identify that area and give a nick or a cut in that area, so that the plasmid cuts open and we can insert the foreign DNA over there. But in some plasmid, it is seen that many restriction sites are present together in one area. Such an area where number of restriction sites are present together is called a polylinker or a multiple cloning site. What is the benefit of having a multiple cloning site or a polylinker? This gives us the option to use different types of restriction enzymes because different restriction sites are present in that area. So, here the right answer is option D, both A and C, because those areas where different types of restriction sites are accumulated are called polylinker, or it can also be called multi-cloning sites, MCS. So, the right answer is D, both A and C. The next question. Statement A, tag polymerase is a temperature resistant DNA polymerase from the bacterium Thermus aequitas. Statement B, it is employed in polymerase chain reaction. The options are, both the statements A and B are correct, both the statements A and B are wrong, statement A is correct and B is wrong, statement A is correct and B is wrong. Statement B is correct and A is wrong. Students, PCR, that is polymerase chain reaction, is a technique to amplify DNA. For any process, if DNA has to be used, quantity of DNA is not enough, then the quantity of DNA can be increased or amplified by the process called PCR or polymerase chain reaction. This was invented by Kary Mullis. Here basically PCR consists of 3 steps. The first one is denaturing. What is denaturing if you ask? The DNA is present in a solution and the temperature is increased to more than 90 degree. That is somewhere around 94 degrees Celsius the temperature will be. This will break the hydrogen bonds between the DNA strands and both the DNA strands will separate. Now each DNA strand will act as a DNA template for the synthesis of the new complementary strands. In this manner, once the new complementary strands are formed along with the existing templates, two molecules of DNA can be obtained. So once denaturation is over, the next one is annealing, primer annealing. Here we use very small DNA segments, or we say oligonucleotide sequences, which will be around 20 nucleotides in length. These are taken and these primers are added at the 3' end of both the templates. When they are added at the 3' end, if one is at this side, the other one will be at the opposite side. Then we have to bring down the temperature to 40 degrees centigrade, and this will go and bind, the primer will go and bind to the DNA. Once this happens, then we start with the next step, which is called extension of the primer. That is, nucleotides of DNA have to be added to increase the primer in length. So again, the temperature has to be raised to 72 degrees centigrade and the nucleotides are added. Now for the polymerization or for addition of the nucleotides, what is required is an enzyme which can withstand the high temperature. Here we do not use a normal DNA like DNA polymerase. Instead, we use a special kind of DNA polymerase which is called tag polymerase. Tag stands for Thermus aquitus. It is a bacteria which lives in very high temperature. So its enzyme will not get denatured at a high temperature. So this particular enzyme is added in the last step, which is called extension of primer. And this particular enzyme start adding the nucleotides to the primer, and a long chain of nucleotide, a long chain of DNA is formed. So in this manner, new strands are formed on both the existing DNA templates. As a result, by the end of one cycle of PCR, we can see that one DNA produces two DNA molecules. So here we can see both the statements A and B are correct. So the option A, both statement A and B are correct, is the right one. The right statement or statements with respect to electroporation in cloning. Options are injecting DNA into the egg, introduction of genetic material through viruses, manipulation of cells by exposing them to a strong electric field. It makes the host competent to take up DNA. Now, if you want to insert a foreign DNA into a cloning vector, or if you want to insert a DNA into a host, the host will generally not take DNA that easily. The simple reason for this is DNA is hydrophilic, whereas the plasma membrane of cells, they are made up of lipids. So they will not allow the DNA to pass into the cell across the membrane. So we have to make the membrane porous so that it can take the DNA into it. So there are different mechanisms to make the plasma membrane porous. One of them is electroporation. Here you can see that slight voltage is given which will make the plasma membrane porous in nature. And when the plasma membrane becomes porous, it is easy for the DNA to enter into the host, or the foreign DNA can be made easily enter into the host. When the plasma membrane is made porous, the bacterial cells are said to be competent. So before sending or before inserting a foreign DNA into a host, the host has to be made competent. That is, it should be made porous for the entry of DNA segments. So here you can see that the option 3 and 4, manipulation of cells by exposing them to a strong electric field, it makes the host cells competent to take up DNA. So you can see the last option, that is 3 and 4, which is option D, is the correct answer. Now let's discuss few more questions based on the applications of biotechnology. Now we can see the application of biotechnology in various fields. For example, in agriculture we are aware of Bt plants, then insect resistant plants, etc. Then in the field of medicine, it is widely used for synthesis of human insulin or humulin. It is used in gene therapy, it is used in molecular diagnosis. Many such biotechnological applications are used these days. So before solving the questions, let me tell you something about GMO. Now, when a gene is inserted into another organism by genetic engineering, that gene is called a transgene. And an organism in which a transgene is present and it expresses it is called a transgenic plant or an animal. In short, a transgenic organism. So a genetically engineered organism, what we call GMO, can also be called a transgenic organism. RNA interference gene interferes with, options are transcription in nematode, translation in nematode, both transcription and translation in nematode, synthesis of rRNA in nematode. Students, before picking the right answer, let me briefly explain you about what is this RNA interference. RNA interference is nothing but silencing of specific messenger RNAs. If there is a gene present, okay, it will produce a messenger RNA, and from the messenger RNA you can see the polypeptide chains are produced. So synthesis of messenger RNA from DNA is called transcription. Then the synthesis of proteins from the messenger RNA is called translation. So the question is at which level it gets blocked. So let me first tell you what is this. In RNA interference, specific messenger RNAs will be silenced or they will be destroyed. When they get destroyed, they will not be in a position to produce the respective proteins. So here, which type of RNAs are silenced? The RNA which is coding for some vital proteins in a nematode. Why? Because nematodes are some of the pests that you see infecting the roots of crop plants. So here, a novel strategy of destroying these nematode is, inside the body of the nematode there will be number of messenger RNAs which will be producing one or the other proteins. If these proteins are very vital for the survival of the nematode, if the protein gets destroyed, ಅಂದರೆ ತುಂಬ ಇಂಪಾರ್ಟೆಂಟ್ ಆಗಿರುವಂಥ ಯಾವುದಾದ್ರೂ ಪ್ರೋಟೀನನ್ನು ಟ್ರಾನ್ಸ್ಲೇಟ್ ಆಗಬೇಕಾದ್ರೆ ಬ್ಲಾಕ್ ಮಾಡಿಬಿಟ್ರೆ ಆ ಪ್ರೋಟೀನ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಲ್ಲ. ಆ ಪ್ರೋಟೀನ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಲಿಲ್ಲ ಅಂತಂದರೆ ನಿಮ್ಯಾಟೋಡ್ ಕೆನಾಟ್ ಸರ್ವೈವ್, ದ ನಿಮ್ಯಾಟೋಡ್ ವಿಲ್ ಡೈ. ಸೊ ದಿಸ್ ಈಸ್ ದ ಪ್ರಿನ್ಸಿಪಲ್ ಬಿಹೈಂಡ್ RNA interference. In eukaryote, it is a common method of cellular defense to prevent the plant from getting infected from different pathogens. So here what happens, how the process actually takes place. We can see that the nematode specific messenger RNAs, they will be coded by a particular gene. So this gene is initially inserted or they are introduced into the host plant. Once these genes goes into the host plant, what happens is it is made to synthesize an antisense and an sense strand. So these two strands which are formed, that is the antisense and the sense strands, both will be complementary. Since they are complementary, they will form complementary base pairing and they will become double stranded RNAs. So these double stranded RNAs are only later going to destroy the messenger RNA of the nematodes. So let me explain this to you with the help of a image. So you can see in the image there are two strand RNAs. From where these two strands are produced, I have already told you people. This is because we are introducing a nematode specific gene into the host plant. Then inside the host plant, complementary to that gene, two RNA strands will be produced, sense and antisense strand. These two, they will undergo complementary base pairing to form the double strand which is shown in the image. Now this double strand, when it goes and binds to an enzyme called dicer, the dicer will cut these double stranded RNAs into small double stranded RNAs. These small double stranded RNAs will be something around 20-21 nucleotides long or in length. So, these double stranded small RNAs are called small interfering RNAs. Similarly, there are other small double stranded RNAs also called microRNAs. So, these microRNA or the siRNA, that is the small interfering RNA, they will go and they will bind to another protein. The name of the protein is argnod. When it goes and binds to this argnod, one of the strand gets fixed on the argnod. That strand is called the guide strand, and the complementary strand which is present along with that gets degraded. So, now with the argnod protein there is only one small strand of RNA present. Now, this particular thing goes and binds with some more proteins and it forms a complex. So, the complex is composed of a argnod protein, few other proteins and a single stranded small RNA strand. Now, since the single stranded strands will have the nucleotides present, they have the capacity to go and bind to any other messenger RNAs. So, the entire complex will go towards the nematode specific RNA and it forms complementary base pairing with the nematode gene. Once it forms the complementary base pairing, the siRNA will direct the argnod protein to start degrading or to destroy the nematode gene. As a result, the nematode specific gene or the messenger RNA will be destroyed. So, when the messenger RNA gets destroyed, it will not be in a position to produce the protein which it had to translate. Okay? So, here what it is blocking? It is blocking translation, production of protein from a messenger RNA. So, once it is blocked, what happens? A vital protein is not produced in the body of the nematode. As a result, the nematode dies. So, this is the basic principle. So, in short, I will tell you, two strand RNA is produced. It goes and binds to a protein called dicer. Dicer will cut it into smaller RNAs called small interfering RNA. These RNAs go and bind to another protein called argnod. Then along with some more proteins, it forms a complex which is called RISC. RISC is nothing but RNA induced silencing complex. This complex later goes towards the messenger RNA because in that complex, from the two strands of the RNA, one strand will be destroyed, only one strand will be there which can go and do complementary base pairing with the nematode messenger RNA. When it goes and binds to it, it will destroy the messenger RNA, as a result of which the messenger RNA will not produce some vital protein. The absence of such a vital protein will not allow the nematode to survive, so the nematode will be killed. So, now these genes, nematode specific genes, are actually introduced into the host plant with the help of agrobacterium. Once the infection is done to the cells, the genes will be introduced into the transgenic plants or the plant becomes transgenic, then it is allowed to grow. So, inside it will produce the double stranded RNAs and the entire process begins. So, the right answer over here is translation in nematode is blocked, that is